thank you all for having us here today. Next slide, please. I am Tracy Thompson, a wildlife veterinarian and the IACUC chair for the National Park Service. And as Scott mentioned, I'll be the moderator for today's virtual panel session. I have no personal, professional, or financial relationship with this educational activity. Next slide, please. To begin with, I wanted to address a potential concern that some of you might raise. Manel is a defined slang or buzzword referring to male-only panels at events. Scott, would you go ahead and hit the bar, please? Thank you. Male-only panels at events such as conferences, and which is used by some to highlight a lack of diversity at these events. As a member of the IACUC 2020 Planning Committee, I can assure you that we did not intend to pull a 180 for this sex bias and research session in creating a womanel. Hit the space bar for me, Scott. Just happened to turn out this way thanks to the very high caliber and renown of the speakers that we have for you today. We do want to recognize, though, that there is a need for a more diverse representation of our community at these sessions. Spacebar, please. And that is a goal for us to work toward that. And we look to you as attendees at this session and at all of our virtual Priminar session that you help us supply the nominations for topics as well as persons to speak on those topics at future sessions and presentations. Particularly in this time, spacebar please, we want to make sure that we recognize current events and the need for increased discussion around racial inequality and justice issues that we're facing in this country. And I'm confident that we as a community can enact change that is meaningful and long lasting if we continue to listen and open up to all of the voices that we contain within our community. Next slide, please. So I am humbled and honored to introduce you to our three presenters today. Our opening speaker will be Dr. Melina Kibbe. Dr. Kibbe is a Colin G. Thomas Distinguished Professor of Surgery and the first female chair of the Department of Surgery at the University of North Carolina. Melina will be speaking broadly about sex bias in animal research, and she is glad to have the opportunity to present this information to those involved in animal care and use committees. Our second speaker will be Dr. Rebecca Shansky, an associate professor at Northeastern University, whose expertise is in studying sex differences and brain function. She is the author of Are Hormones a Female Problem for Animal Research?, published in Science. Becca will speak to issues of sex bias in research with a particular focus on rodents and neuroscience research. Our final speaker today will be Dr. Natalie Tronson. Dr. Tronson is an assistant professor of psychology at the University of Michigan, whose research focuses on the molecular mechanisms of learning and memory, the internal and external factors that can lead to changes in memory, and the role of memory dysregulation in psychiatric and neurological disorders, including post-traumatic stress disorder and post-operative cognitive decline. Natalie will further refine the impacts of sex bias as seen in behavioral studies. And I'd like to now turn this over to Dr. Kibbe.
Wonderful. So Scott, if you'll stop sharing and then I can share my slides.
Yeah, I believe, Lena, you can go ahead and share and then we'll.
Perfect.
Great.
There we go. Wonderful. So thank you so much for that introduction. And also thank you so much for addressing the issue of the fact that we're a wool mantle and went the other direction. We did have a very healthy discussion on that. So I'm glad you addressed it because it clearly was not our intention, especially on a panel that is here to talk about sex bias. All right, so with respect to this talk, I am going to give a more broad introduction to this topic. I have no pertinent financial disclosures as it pertains to the material of this talk. So let me start by asking everyone, are these two individuals similar? I think we would all agree no. Are these two individuals similar? I think we would agree they are not. So that brings me to a question that I'm going to pose several times over. Then why do we treat men and women the same? And taking it one step further, why do we treat men and women with the same drug for a disease? We know that men and women are not the same. So we know that there are a lot of sex-based differences. Men and women can manifest diseases differently. They can experience illnesses differently. They can respond to drugs differently, metabolize drugs differently, respond to devices differently. Think about hip replacement. We certainly know that a female hip is very different from a male hip. And also they can have different outcomes following medical and surgical therapies. Now, here's a slide of just some common diseases that have known sex and gender differences. Now, I like to point out at this point that I am referring to sex throughout my talk, our chromosomal differences. I'm not referring to gender, the phenotype by which we display ourselves. So just remember that. Now, I happen to be a vascular surgeon. So if you look at this list of diseases that have known sex and gender differences, I kind of fall into this category of heart disease. Let me show you a few facts. Now, this is a graph from the American Heart Association, and many on this panel and this call may already be familiar with this. So this is the incidence of cardiovascular disease as we age and by sex. So I think most people have an appreciation that the incidence of cardiovascular disease is actually higher in men than in women. But did all of you realize that more women are dying from the disease than men until just recently? This is what most people don't realize. That's a major difference between the sexes right there. And if you look at some of the common medications that I prescribe in my own field, let's just review a few of those. Aspirin. This is like vitamin A to me as a vascular surgeon. But aspirin, there's a big difference between the sexes. It is actually recommended for prophylaxis for men when they hit the age of 50 for prophylaxis for cardiovascular disease. It's actually contraindicated for women. It is not recommended for women and in fact has more adverse problems. Statins, more beneficial in men for primary prevention, but greater risk of adverse reactions in women. Angiotensin converting enzyme inhibitors, more adverse effects in women. Digoxin, increased mortality in women. And warfarin, usually women tend to require lower doses than men. Now, these are just some common medications in my own area. Imagine the differences in all the different areas of medicine. So it takes me back to that question. Then why are men and women given the same drug for a disease? And why are men and women given the same dose of the drug? Can any of you think of a drug that is dosed differently based on sex? You think about that. Think of any medications you might be taking or anybody you know. You're going to be hard pressed to come up with one. Well, we have one now, Ambien. So Ambien is an interesting story. Ambien is a sleep-inducing medication. And what happened with Ambien is it was originally approved by the FDA back in the 90s. And it was approved, of course, for one dose for both sexes. Well, what happened several years ago is that they were starting to notice more reports of women who, after taking the drug, the next day usually, were getting into automobile accidents. So in studying this, they realized that women metabolize the drug more slowly than men. So the next day, they still had this drug in their system. That's a problem. Again, this is just a sleep-inducing medication. And this led the FDA for the first time to issue a label change on the medication. So now, Ambien, for women, it's recommended they take half the dose, 5 milligrams, versus the 10 for men. Now again, this is just one drug. Imagine all the other drugs that this probably needs to happen to. It's staggering when you think about it. So that takes me to the root of the problem. So here's the typical pipeline for drug discovery and development. So typically, researchers will start by investigating a therapy down here at the cell level. And then as this is successful, it will start being translated to the animal level, hence our conference, the IACUC. And then if it's successful here, it will be translated further into human clinical trials. Now, I will pose that one, not the only, but one of the roots of the problem is that we've had a focus on the human aspect of this research. Back in 1993, we had the Revitalization Act, which was signed into law. And for those who are old enough to remember this, it requires the enrollment of women and minorities in clinical trials funded by the NIH. But traditionally, there has been absolutely no focus on equally studying both sexes at the early stages of this pipeline. None. So that is a problem. So when I was at Northwestern, we, as part of a group of individuals, decided to take a look at this. So again, I'm a vascular surgeon, so we decided to look and see if there was sex bias at these earlier stages of research, the biomedical research using cells and animals. And so we decided to look at our own area of surgery, and we hypothesized that sex bias would exist in surgical biomedical research. So what we did is we went to 5 of our common broad surgery journals, so not specialty specific, and we basically examined every single publication in 2011 and 2012. And we abstracted different information, including if animals were used, the sex of the animals, how many, if cells were used, the sex of the cells, etc. So this is what we found. We came up with, out of these 5 journals, a little over 2,000 articles. After excluding the humans, we were left with 618 articles that used cells or animals. All right, so on the animals, one of the findings that I was not expecting to see, very relevant for this conference, 22% of the publications did not state the sex of the animal researched. That's a problem. Now, of the 78% of published manuscripts, and these are peer-reviewed published manuscripts, of the 78% that did state the sex, 80% studied only male animals. 17% female, only female, and a tiny little 3% actually studied both sexes. Now, what about cell research? So cell research had a different issue. Here, 76% of the publications did not state the sex of the cell being studied. Now, of the 24% that did state the sex, same pattern. 71% male only, 21% female only, and a little 7% actually studied both sexes. So then we said, well, what happens if we look at those publications that were studying diseases prevalent in females? So in our surgical literature, we were able to very easily look at cardiovascular and thyroid, as these are diseases prevalent in women. Well, 44% of those publications did not state the sex studied. And of those that did, even worse, 88% male-only studies. And then we did one more thing. After seeing this data, we then asked ourselves, well, have we been getting better over time with this practice, worse over time, or has it stayed the same? So we went back 2 earlier decades and abstracted all the manuscripts in those earlier years. And this is what we found. We've actually been getting worse over time with studying male-only studies. All right, so what are the implications of this sex bias research? You're going to hear more from the other speakers, but I'll just tell you this, and I know this is a little grandiose, but you're going to get drugs that are developed that just may be ineffective for women. If you are developing them and studying them mostly in males, this is what could happen. You also could have drugs that are developed that'll have unintended side effects in women. Look at the drugs that I showed you in my own specialty and how many of them had side effects in women. And then drugs that show no efficacy in men might be dropped and not pursued, but they could have efficacy in women, but we would never know. So these are some of the many implications of this sex bias research. And then this is my most important slide. Did many of you realize that 80% of the drugs that are removed from the market by the FDA have been removed because of serious adverse reactions in women? 80%. It leads to lots of stories like this, how bad medicine dismisses and misdiagnoses women's symptoms. And we even get things like this. The rats are facing sex bias as well. So how do we fix this problem? This is a really good question. So I would say that we need increased awareness. And that's why I was thrilled to learn that you were having this panel session today at this virtual IACUC conference. I think it's really important to increase awareness with the investigators. There's so many times where I will give this talk to a room of researchers and they haven't really thought about this issue. And you're gonna see, I was one of those. You'll see that in a minute. There's physicians. So oftentimes physicians don't think about this also. And so increasing awareness in the physicians who are prescribing these medications is important. And then there's the patients. Oh my gosh. When I give this talk to lay audiences, it is guaranteed that as soon as I'm done, a question from the audience is, but Dr. Kibbe, I don't understand. If my doctor prescribed that medication, I just assumed it was tested in both sexes. And that's because of the implicit trust in the medical community. So I think it's really, really important to increase awareness. Basically in May, the NIH announced this, their plans to develop a new policy that would require investigators to report their plans for the balance of male and female cells in animals in all preclinical studies. This was so tremendously exciting. And I can just tell you, we were waiting and waiting and waiting. Finally, a year and a half later, the NIH came out with this, implementing rigor and transparency. And I'm sure many who have joined this call are aware of this. And so basically it requires all grant applications going to the NIH to include sex as a biological variable in the grant applications. And it is now a score deriving factor. So I am super happy about this. Now journals, I would say journals have a really important role to play. And I just showed you a little bit of that data. I personally believe that journals should require all authors to provide a description of the sex studied of the cell, the animal, or the human. And if only one sex is studied, then simply require a single sex justification statement. And I really think this should be a requirement of all journals. I'm happy to report that in July of 18, all of the surgery journal editors got together and we jointly published a statement in support of this across a whole bunch of surgery journals. So one journal at a time, we're getting there. The FDA. Now, I think the FDA has a very big role to play here. So after this story aired in February, and then in May, when the NIH released their statement, this came out from the FDA action plan to enhance the collection and availability of demographic subgroup data. Okay, this was a good start, but this definitely was not enough. So then a couple years later, they came out with this. The FDA came out with the Women's Health Research Roadmap. Look, they have 7 priority goals. Priority goal number 1, advance safety and efficacy. Advance the safety and efficacy and reduce the toxicity of FDA-regulated products used by women. I thought that was their job. I'm just saying. If I had more time, I would expand. Anyhow, industry-funded research. Now, currently, there is no mandate or policy for industry-sponsored clinical trials. When I mentioned the Revitalization Act, that only applies to NIH-funded clinical research. So the only thing that industry-funded research is held to is basically the FDA when they're submitting a new drug application. I personally believe that the same guidelines that apply to government-funded research should apply to all clinical trials. Because guess what? 90% of clinical trials are industry-sponsored, not NIH. And then lastly, I'll just say that I do think there's a role for the government. I was so thrilled after the 60-minute story aired and the NIH came out with their statement. Congressman Jim Cooper came out with Research for All, Act of 2015, and this has requirements for both the FDA and the NIH for studying both sexes equally. So when people ask me what they can do, I say, write to your congressman, have them support this. So in summary, hopefully I've conveyed that sex does matter, and this is the topic of our panel. Research is fraught with sex bias. While I showed you data on the surgical literature, trust me, it is the same throughout most all of our disciplines. Of the manuscripts that stated the data of the animal or the cell, about 80% were studying males only. It's a problem. There's a great need for increased awareness, and there is definitely need for more policy change. With that, I would like to thank you very much, and I am looking forward to our panel discussion. Thank you.
Becca, we'll go ahead and turn it over to you.
Okay, great. One moment. Okay, that was a great intro for what I'm going to talk about. So thank you, Melina. So I'm going to narrow things down and talk a little bit about sex bias in neuroscience research, as well as how this concept that Melina brought up already, that that male mice became kind of the default research subject in labs in lots of different fields. And so neuroscience, it should go without saying, is pretty awesome. It helps us understand how the brain works. And of course, we all have brains, pretty sure. And what you're looking at here is not a super zoom in on a Monet or a pointillism kind of painting, but this is actually a photograph taken on a microscope of a mouse brain. This mouse has been genetically engineered so that its neurons in its brain all express different kinds of colors, and that can help us understand how different parts of the brain are connected. And the mouse has a very clever name, which is Brainbow. Usually very popular. And we can also get really amazing images of human brains too using diffusion tensor imaging. And so understanding how the brain works is, of course, very, very important. There are tons and tons of things that we need to understand about just basic brain function, as well as all the ways in which brains can dysfunction. But as you have already heard in the context of cardiovascular research, neuroscientists also have kind of an embarrassing secret, which is that what we think we know about the brain, we really only know about male brains. And that's because, like some of the other fields we've already heard about, neuroscientists have been studying male animals about 6 times as much as they study female. So you can see that this proportion is very similar to what you heard from Melina's. And so this is a public health problem, pure and simple. And I think a lot of that was outlined really nicely in the last talk. With respect to issue or disorders that affect the brain, we know that almost any mental illness or neurological disorder you could name occurs in both men and women, but they often occur at different rates. And so what this chart is showing you is the rates of several mental illnesses, with the disorders that are most affecting women at the top. And so those include things like PTSD, panic disorder, anxiety disorder, major depressive disorder, whereas the disorders that affect men most prevalently are shown at the bottom. And so these discrepancies, you know, there are probably some cultural, sociological factors that contribute to those discrepancies, but that's not all of it. You can certainly do, you know, sort of the analyses to take those factors out of the equation. So we think that there are probably some neurobiological reasons why men and women would be differentially susceptible to these kinds of disorders, but we're not going to know where that susceptibility comes from if we're only studying male brains. And so for my specific interest lies in some of the disorders that are more prevalent in women. And what you can see about these top 4 is that they are all related in some way to stress. Either their symptoms are characterized by aberrant stress responses, or they are caused by experiencing a stressful event or a traumatic event as well. So there's lots of sex differences in the way we understand how the brain processes stress, but we still don't know enough. So to get at some of these questions, say why don't neuroscientists want to study females? You can ask anyone, and the answer is perfectly clear. And Colbert got at that a little bit. The answer is hormones. And so what you're looking at here on the right is a graph showing the hormone fluctuations in a rat versus the hormone fluctuations that you see in a female human. And so what you can see is that in both cases, there are peaks of hormones like estradiol, progesterone, luteinizing hormone, and then there are also lower levels as well. And so the rationale for not studying females is, well, some of your animals are going to have high levels of estradiol and some of them are going to have low levels of estradiol, and that's just clearly going to mess up all my data, right? So neuroscientists think that if they study females, their data is going to be messy. And of course, we're scientists. We don't like things to be messy. We want really clean data because we're searching for the one thing that is true in all of our experiments. And it felt like these hormones were going to be kind of a nuisance. And so basically the rationale was always just, well, we're just not going to study females because the estrous cycle is going to get in the way of discovering the truth about the brain. And so that was just sort of accepted for a really long time as a perfectly normal reason for leaving out half the population. And so I have been thinking about that, and it sort of reminded me of something that we've maybe seen in popular culture. This is a really old internet meme, which maybe some of you have seen before. You can tell by the old Mac font. And, you know, people laugh at this because I think a lot of people think it's true. They're like, oh, men are so simple and women are so complicated. And it occurred to me that was sort of the line we were hearing from neuroscientists as well, is that studying female mice was as complicated as it was to understand what it meant to be a woman. And so I started to think about where this idea came from in the first place. And so, you know, of course, we've seen this in you know, just general news and popular culture, this idea that women are hormonal, they're emotional, and that makes them essentially unpredictable, and that this is somehow overlaid on top of, you know, real humanness. And so again, I saw these parallels and I started to dig a little bit deeper into some of the history of where these stereotypes came from. And so what I discovered is that we can actually blame Victorian era neurobiologists, or not neurobiologists, just biologists. People were not as specialized back then. And essentially, around Victorian times, it became slightly un-PC to simply say that women were an inferior version of men, which had basically been the party line up until then. So they had to kind of restructure their narratives in order to say, okay, women, you have your own qualities that are different from men, but the qualities they essentially gave us were sensitivity, emotionality, instability. And so all these kinds of qualities essentially allowed them to argue that women were unfit to hold positions of leadership in society, and so thereby they preserved the patriarchy, and this narrative has perpetuated through over 100 years. Basically, people still hold these opinions, and we have essentially transferred those opinions onto our animals. And so once again, the neuroscientists' line is that data from females will be more variable than the data from males. But no one actually ever asked, is this true based on real data? And until this decade, in 2014, what is now kind of a canonical paper entitled "Female Mice Are Liberated for Inclusion in Neuroscience and Biomedical Research," where they essentially did an analysis of lots and lots and lots of data from all different kinds of neuroscience papers, and they found that if they looked at the actual variability of the data, there was no difference between the data they collected from males versus the data they collected from females. So the variability argument is essentially out the window. This was in mouse research. This same thing was repeated a couple years later looking at rat research, same exact findings. So there's no difference in the variability of the data that you collect from females versus males. It is possible that the estrous cycle contributes to the variability you see in females, but that doesn't allow us to understand what the variability we might see in males is, and it's essentially been accepted that whatever variability you see in males is normal and totally fine in terms of experimental analysis. So we have another little secret. Males have hormones too. I'm sure most people in this audience know that, but they can also vary, and they vary according to the social dominance hierarchies that mice put in a cage together will create when they're being housed together. And so animals, if you have a group of maybe 4 or 5 male mice in a cage, someone's going to come out after a couple of days as the alpha male. That one could have up to 10 times higher testosterone levels than its subordinates in the cage. And so this is the paper from the 80s, by the way. We've known this for a long time. Somehow it hasn't really interrupted neuroscience research, despite males having pesky variable hormones as well. And so I just want to talk a little bit about what happens when we have been using males as the default research subject. What do we miss? How do we identify the right symptoms to treat if we're not even knowing what we're supposed to be looking for? And so I'm giving you an example here from my lab. This is a rat who's been trained to fear this tone that just came on. You can't hear it, but you can see how it responded. And so it's going to just go kind of pick this corner to sit in and engage in what's called a freezing response. And this freezing response has been essentially the single way that neuroscientists measure learned fear in the lab forever. But what we have found in my lab, missed that little animation, is that females actually can do a lot more different things when they get scared. And so here's a female. You can see she's exploring. That same tone is going to come on. And so she does a little spin around. She runs to the other side. So some similarities in what we saw with the males, but she's also doing some other kind of vigilance behavior. She does a little bit of this freezing behavior that we saw in the males, but there is more that happens. And you can see that already now she's kind of slowly trying to see if it might be safe to emerge. And so the question becomes, what should we be looking for when we want to try and treat these different symptoms? We need to know what the symptoms are in males and females. And so as Melina mentioned, many of the drugs that we use today for everyone were developed using only male animals, so we might not be treating the right symptoms in women. And so we need to be able to study behaviorally the different kinds of things that, behaviors that females use. And as you also heard, in 2016, the NIH began requiring everyone to use both sexes, but there is not a lot of accountability right now beyond the grant review process. And so what I think is going to have to happen is that we need, in order for people to do it the right way, we need education in terms of how to design unbiased experiments that don't rely on male-defined outcomes, as well as accountability from the NIH itself, as well as journals, as Melina mentioned. So just a couple of quick take-home messages. So we need to consider the influence of societal stereotypes on how we view research in males versus females. I know they're rats and mice, but we can inadvertently anthropomorphize in terms of how we're interpreting what we find in them. Hormones are important for both sexes, but they are not more important in one versus the other. And we need to question the history of our models in the first place. If they were developed and validated in males, they might not tell us the same thing in females. So with that, I will say thank you. Here's my gang. And I'm also looking forward to our Q&A.
Great, we'll go ahead and have Natalie take over from here.
Yes. Sorry, I'm just closed something unintentionally. Sorry about that. So what I am going to be doing while I'm opening it, I will tell you, is I am going to be doing a specific example of all of these issues that Melina and Becca have been talking about. And specifically, whoops, we'll go back to the beginning. Specifically, I'm going to be talking about sex differences in learning and memory, or a cautionary tale of what we miss when we focus on male bias studies. And I have no conflicts of interest to declare. So as we've heard now a few times, most of the research in biology, in neurobiology, has focused on males, and in neurobiology of learning and memory, this looks particularly bad. It's not 20%, it's less than 2% of studies have focused on females or have included females at all. Now, this was work up until 2010, so maybe things have changed a little bit. But what this means for us now and still is that the behavioral responses, as Becca just mentioned, for what we think about as learning are defined in males. The molecular mechanisms, the neural networks, the circuits have all been defined in males. And even with sex as a biological variable, and this is data that just came out last week in eLife from Teresa Woodruff's lab, and this is just from the last 10 years, this is for all of neuroscience. When we look at studies that include females, that's increased a little bit, that's great. But when we look at studies that analyze by sex, that look and see, are there actually sex differences there? That hasn't increased at all in the past 10 years. So that's disappointing. Hopefully that continues to change. So there's a specific logic for testing males. We've heard about it a little bit. I'm going to talk about it in terms of memory here. So we assume that males and females are more similar than different, and learning and memory have nothing to do with reproduction. So why would there be differences between the sexes? And memory is fundamental to survival. We have to learn where dangerous things are in order to avoid it. We need to remember where food is and who our people are. So if it's fundamental for survival for both sexes, why would it be different between the sex? And finally, as we've heard, females have pesky hormones, so we should just ignore them, focus on males, and then generalize to females too. So this is hugely problematic. And one of the reasons it's problematic is that we know that there are sex differences in learning and memory. We see this as sex or gender differences in disorders of memory. So we see twice as many women than men developing Alzheimer's disease and faster progression in women than men. This is not just because women live longer. We see almost threefold more women than men developing post-traumatic stress disorder, which is about excessive memory for traumatic events. And in converse, diseases like Parkinson's disease show faster cognitive decline in men than women. So even when you have both men and women with the disorder, you see sex differences in progression of the disease. So this might be partly about the disease progression, and it probably is, but we also see sex and gender differences in memory. So verbal memory, women show better verbal memory than men. We don't know if that's a biological sex difference or if that's a culturally gender-defined difference. It could be either. It's probably both. We see differences in both mice and humans, where women and female mice are better at object recognition than males. And in spatial memory, where males are better than females and men are better than women. We also see some differences in fear-associated memory, and that's what I'm going to focus on today. But I first want to say we should think about what we mean when we talk about sex differences in memory as well. We usually talk about this as amount, right? As unidimensional, men are better than women, or males are better than females, or vice versa. Females are better than males. That's just what I said in the last slide. But we should also be thinking about this in a multidimensional way. We might see differences in terms of what information we learn about in the environment, not whether we have a better memory or not, but what we're thinking about, what we're looking at, in different behavioral responses, and in what molecular mechanisms, neural circuits, and what memory systems we're using. All of these things can be different, even if we see very similar performance on the task. And again, the problem here is that behavioral responses, memory mechanisms, molecular mechanisms have all been defined in males. So if females use different strategies or different mechanisms, it might look like worse performance. We might end up saying females are worse than males at this task, even if it's not about ability, it's about strategy. And there's an old example of this, and this is commonly used. We've probably all heard before today that men are better at spatial memory and spatial tasks than women. And in terms of spatial memory, one of the tasks we use is this Morris water maze for rodents. And we use a virtual version of this in humans as well. In rodents, what we do here is we have a pool with a platform that they can escape on, and we let the animals swim around until they find the platform. Now, where that is in the room is defined not by anything cueing the platform itself, but by objects around the room. So they're able to map the room and then define learn where that platform is in that room. And males and females can both learn this, but males learn it faster than females, and they have a better memory when we retest them again. The problem is that women don't walk around in the world lost all the time. At least I don't see other women walking around lost in the time. And our spatial environments are a lot more dense and complex than just having these distal cues around the outside of our world. So what happens if we look at other kinds of spatial cues? So research that has used this same task, but instead of just these distal cues, that have added landmark cues or closer cues, local spatial cues, slopes of the ground, all sorts of things, have shown that males in rodents and men in humans are biased towards these distal spatial cues. Female animals and women are biased towards using local spatial cues. So you can shift who looks better at this memory task depending on what kinds of cues are available during test. So it's not that men are better than women at spatial tasks, but there are sex differences in what information is preferred. Now, I also want to say this, we generalize males and females as if these are two dichotomous populations. We shouldn't really be thinking about it like that. There are some men who are going to use more female-like strategies and women that use more male-like strategies, and a whole range spectrum in between. But in general, we can see these divergences in the behaviors and the strategies. So what about fear conditioning? I'm going to be talking about context fear conditioning here, where animals learn that a place, and this is a little experimental chamber, that this specific place is dangerous. And then they show these freezing responses or other kinds of fear responses. And I'm going to be talking about freezing in this context. We use this as an experimental model for studying neurobiological mechanisms of memory formation. It's great for that. Humans and lab animals learn this in a single session. We see very similar circuits between humans and rodents as well. We can also use this as a model for learning about trauma, and in doing that, we can start thinking about mechanisms of things like post-traumatic stress disorder and treatments for fear and anxiety related disorders. And if you've ever heard of exposure therapy, this is a type of therapy, a type of treatment that's come directly out of animal work on decreasing fear-related behaviors in learning tasks. So are there sex differences in context fear conditioning? And of course, yes, because otherwise I wouldn't be talking about it. But what are those differences? Well, traditionally, the answer to that is that females don't learn context fear conditioning very well because females are bad at spatial learning and bad at context learning. And this idea is based on a relatively small number of studies from a few labs, but it's persistent and it's true still in many labs. But it seems unlikely that females can't learn that a place is dangerous. Surely it's important for females and males to learn to avoid dangerous places. So what happens if we think about it in a different way? What happens if we think about it in the terms of what information are females using and males are using? What are the behavioral responses in males and females? What types of systems and circuits are males and females using that might be similar or different? Another reason to question this is that whether you see this difference in whether females can learn this task depends very much on the lab, the animal strain that's being used, and the precise protocols. And this precise protocols is something we come back to all the time when we're talking about learning and memory, because we define what animals are learning by how we're studying it and what protocols we have in place. So this is data from my lab. It's not that females can't do this task. In my lab, we see over many, many experiments, we see very similar performance in both. And I know nobody's going to ask this because of Becca's talk, but in case you do, we also see no differences across the estrus cycle. So what we did here is we trained the animals in context A, and this is a standard experimental box. And then, like with the spatial memory example I just gave, we messed around a little bit with what information was then available during retrieval. So usually when we're doing context fear conditioning, we train them in one context and we put them back and see if they show fear to that context. Here we did that, but we also looked at a similar context, I'm going to call this B, where we changed the shape a little bit, but the floors are the same, the smells are the same, the light was different, but the sound in the background was the same. And then we took another context, context C, and changed everything we possibly could. The walls are different, they were patterned, they were curved, the floors were smooth, it smelled different, everything we could change, we changed. So what do males and females look like then? So first of all, both males and females learned just fine. And in fact, if anything, females were freezing more than males in this series of experiments. And both males and females learned about context, because when we put them in this very different context, this third context, they stopped showing so much fear. So they knew that the place where they were trained was dangerous and this very different place was okay. But when we looked at the similar context, we saw something very different. And this is this middle bar here. Females still showed a lot of fear behaviors to this context, whereas males started acting as if this was a neutral context. So either this means that females are unable to use detailed context information, or perhaps males and females are responding differently to ambiguity. When you have some ambiguity about danger, you can either be like, okay, I don't know if this is safe or dangerous, I'm going to assume it's dangerous. Or you can be like, whatever, it's probably fine. And maybe what we're seeing here is a difference in those sort of risk strategies. And that's something that's common in other sex differences research in other kinds of anxiety tasks. So we can also look at this in terms of what brain regions are activated during context fear conditioning and during retrieval. And I'm particularly interested in hippocampus, which we know is important for memory in both males and females. It's important for spatial memory. It's important for context fear conditioning. And the amygdala, which is important for emotion and fear and all forms of fear conditioning. So I'm just looking at how much this is activated. These regions are activated in males and females. And when we look right after the learning task, So this is while the memory is being stored in the brain. Males and females look pretty much identical. They both have strong activation in both hippocampus and the amygdala. Great, we know that. We think that is what should be happening. Fantastic. But when we look during retrieval, we see something different. What we see during retrieval is that males are relying on the hippocampus. They're more strongly activating the hippocampal system, whereas females are more strongly activating the amygdala. So I don't know entirely what this means in terms of the behavioral results. We don't know that yet, but it does start to talk about when we're thinking about sex differences, it's not just about who's better, who's got a stronger response. It's also about what they're doing and what brain regions are being activated. So males and females are biased towards different brain regions. And during ambiguity, females may use a more risk-averse strategy. But more importantly, what this means is that theories and tasks developed in male animals don't always hold true for females. And that's true in this sort of very basic neuroscience field of learning and memory, as it is for Melina's work in surgery. It's not enough to note that there are sex differences. We also need to be able to do the work, which we haven't done yet, to understand what females are doing. So coming back to vulnerability and memory disorders, what do differences in learning and memory mean for this? Well, if we focus on similarities between males and females, then we start to talk about sex differences in vulnerabilities or gender differences in vulnerabilities as weaknesses, which isn't actually very helpful for developing treatments. If we focus on differences in mechanism, in strategy, whatever we want to think about, then we can start to ask about what leads to that vulnerability, and more importantly, how can we target treatments for sex-specific approaches and having efficacious treatments for both males and females. So coming full circle back to the very beginning, this also means that diversity in science matters. So one of the reasons that we have ended up being a womanal here is that we're the people that are looking at females, that are including sex in our studies, and we think it's important and interesting. And the people that are doing and funding the research in general determine what questions are interesting. We're the ones that develop the behavioral tasks, the theoretical models, and the assumptions that underlie this. And in this case, we're talking about how studying one sex, largely because we've traditionally had a male-dominated workforce. And now when we have more women coming in and starting to think about, well, what are we missing here? What do females do? What do women do? Now we have massively more women studying sex differences in these questions than men. But what else are we missing? When we increase diversity in our workforce, we're going to open questions that we haven't thought about before because of our own blind spots. And we're going to discover more biases, probably some very embarrassing, problematic biases as well as we move forward. And with that, I would like to thank my people, particularly Ashley, who did all of this work. And we can move on to questions for everybody. Thank you.
Thank you so much, everybody. And we definitely have had some questions coming in, some great questions. I know Melina's been able to answer quite a few of them since they were initially coming in for her during her talk. But I did want to see if there's any other questions that folks want to ask. They can submit those through the Q&A section. But while we're waiting for that, I did want to ask one to the whole panel, specifically the last question that we had entered on what suggestions you three might have for how the IACUC could have a positive influence on researchers as far as addressing sex bias in research, especially things the IACUC can do that won't be viewed by researchers as being bossy and trying to tell them how to do their science.
Can I tackle that one? I already responded to that question, which is excellent for this panel. So I actually personally think that the members of the IACUC are critical to this issue because all of the research for animals is going through the IACUC. So by doing something just as simple as trying to add a checkbox to your institutional form asking the investigators the question, are you studying both sexes? Or put, you know, how many are males, how many are females? By just asking that question, it will make the investigators take pause and think about it. And then just like I said for journals, if the PI, if the investigator is only going to study one sex, ask for a justification statement for the single sex study. Those are things that do fall under the IACUC purview, and you can do. These are things that we were doing at Northwestern before I left. So it's critical, and this is the right audience to tackle this issue.
One other simple sort of management side issue is having ordering forms where we can order both sexes in the same order, which sounds so trivial and would be so helpful. And working with and having different strategies. There are a variety of different strategies for best practices for whether males and females should be housed in the same room or different room, or depending on the studies that are being done. So being able to, knowing what those are and being able to be open to those conversations about housing and handling and those kinds of issues would be helpful too.
Becca, did you want to weigh in on anything differently?
No, I was thinking about the housing thing as well. I mean, that seems that's right in everybody's wheelhouse here. I think there is a lot of concern. So obviously, I think one of the biggest demands, I would imagine, in any vivarium is space and number of rooms that people can house their animals in. And a lot of people might say, and by people, I mean the scientists, might say, oh, well, I can't add females, sorry, because I don't have a separate room in which to house them. And the reality is that it is totally fine to house males and females together. I've been doing it for 20 years, and you know, my experiments work. So I think the argument that they have to be housed separately, you know, unless you're studying reproductive something, maybe, but the reality is, like, in the real world, and probably over at Charles River or wherever the animals came from, they were probably not housed separately. And so I think the idea that they're, you know, you're again gonna like mess up your data if the males and females can smell each other is probably not really founded in a whole lot of reality.
Yeah, and if you send them to Jill Becker, she'll say that you should be housing females in the same room as males, and to not do so is problematic.
Tracy, can I answer that next question that came?
Please, I was going to read it out for the group, but
yeah, so let me, because I looked at this exact question. So there's a question from the audience of, are there any studies that show if female researchers are actually more likely to choose female sex for their research, or do you think they often still choose males for the same reason? So I went back to those 2011 and 12 publications and wanted to explore this very question. We actually did not find an association of female investigators versus male investigators using any more or less of one sex. But we did find two interesting findings. When it came to authorship, we did find that senior authors who were women more often published with first authors who were women. So women-women pairing, which I found more than would be statistically predicted. So that was an interesting finding. And then akin to an earlier question that was really important, I found something that made me super happy. We looked at the subsequent citations of those papers. And what we found is that the manuscripts that had sex inclusion, so the ones that studied both sexes, analyzed the data by sex, and did sex-based reporting, on average, drew 3 times more citations than the sex-biased manuscripts. So that means that our scientific community, thankfully, is recognizing sex-inclusive research is more impactful. So that made me happy.
Great. In light of that question, we have another one about, have you seen any research conducted with using both sexes translate into human clinical trials?
Well, so human clinical trials, the issue with human clinical trials is we're doing a good job including, and I didn't show any of this data for the sake of time, It's in my longer version of the talk that I've put the link out. The issue with human clinical research is a little different. So we're doing a good job of at least including both sexes, but what we're doing a bad job with is we're not doing sex-based reporting of the data, and we're not doing a good job of trying to match the inclusion of the sexes, meaning roughly equal numbers of males and females, or numbers in males and females that match the prevalence of the disease. So those are the big areas. For example, that Ambien, when we pulled the new drug application to the FDA, guess what? They studied 16 females and 49 males. And that is a pattern that persists to this day on clinical trials. More women need to be enrolled in clinical trials.
Great, thank you.
I would also,
Rebecca?
Oh yeah, I would just add that in terms of the timing from when the NIH introduced this mandate, so they introduced it in 2016, which means that the people who adhered to it in the grant applications that they wrote didn't even get their money until 2017. So really a long way off from clinical trials that reflect people actually responding to the NIH mandate. And so if there are clinical trials that result from research that was conducted in animals in both sexes, they were probably very small. I mean, I just think there wasn't a lot out there for clinical trials that are currently in process to pull from. But maybe in like 10 years, we'll hopefully have a better answer.
Excellent point. Thank you. I have another question here for you about COVID-19 and whether or not there's any historic sex preference in infectious disease models. I know that's not any of your guys' particular area of expertise, but would you speak to that?
So the only, again, this isn't our, my field. The one piece of information I have for you is that it's my understanding that in immunology, the culture is that they use female mice more than male mice because they don't fight each other. And
because they have a better immune response.
Okay, so, yes.
Yeah.
And also that, so that's the closest thing I have to an answer on that, and that side. But there does seem to be, I'm sure some of you have seen the news articles, it does seem to be COVID prefers men. So we're learning a little bit about that too.
From the immunology side, not so much from the infectious disease side, but from the immune response side, there are precedents for some of the strong immune response cytokine storm correlates of these diseases to have worse outcomes in men. There's not a lot on it though, and you'd think there would be, but there are certainly differences in immunology between males and females, and that's probably better studied in that field than most, but I don't know about the infectious disease side. Do you, Melina?
No, there definitely are sex-based differences in both immunology and infectious disease. I think for the sake of time, because I see that Scott came on and also not my area, but they do exist. There are plenty.
I'm just checking in with our premier coordinator since we're at 4:15 your time.
Yep.
Just want to see if we need to turn it over and capture questions later.
Yeah, so everyone knows we're going to be exporting all of the questions out of Zoom and supplying them to the panelists, so they'll have the opportunity to answer any that they didn't have time to answer if they wanted to answer it in any depth. And you can find that we would be posting this for this session and for any other session on the site as additional resources.
Thank you to our wonderful panelists. Really great discussion and excellent questions coming in, and look forward to continuing the discussion, I think, on reducing the bias in our research animal studies. So thank you all very much.
Thank you.
Thank you.
Thank you. Thank you for having this panel.
You're welcome. And thanks to everybody for developing and presenting the session. And I also want to thank everyone on the line for joining us. I know it could be a challenge to sit all day on the computer, but hopefully you found the panels and breakout sessions beneficial. Okay, I'm going to stop recording, and then I'm going to share the next information on the next panel.